Hello everyone. Welcome to part 10 of the Biology Crash Course for Bioinformatics video series. So in this tutorial, we are going to talk about evolution and phylogenetics. So this is a very important topic because a lot of people work with evolution and phylogenetics, how these trees are constructed and things like that. And it's a big area of bioinformatics research. So I think if you want to do that kind of research, then this video is a very good start for you so that you can understand when you read the papers. So first of all, let's go to the phylogenetic tree. So let's not complicate things first. Let's just look at a family tree. So everyone understands the family tree, right? So assume that this is you, the one who is watching the video. This is you. Maybe you have some siblings and both of you obviously come from the same parent, right? Okay. Now, obviously your parent also has a parent who is known as the grandparent, and your parent, maybe this is your mother. Your mother may have a sister. So that person is your aunt, right? And that aunt can have maybe two children, and both of them are your cousins, right? So this is your family tree. So if you look at the family tree, there are two interesting things we want to actually understand here. Number one is you are actually the closest to your sibling, right? Genetically. I'm saying genetically, and even I would say to some extent in characteristics, you are very similar to your siblings. Both you and your siblings, they are actually equally far apart from your cousins, right? From your cousins. And both, like your cousins and you, your siblings, you share a common ancestor, which is the grandparent, right? So that is the phylogenetic tree. So it sort of shows like which leaves are closer to each other. The cousins are equally close. You and your sibling is equally close. And both the cousins are equally far away from you and your siblings because all of you come from the same ancestor, right? Who is the grandparent. So let's now look at a phylogenetic tree, an actual tree. So this is an actual tree. I mean, obviously the trees are much bigger, but just to give an example, I'm just showing very small part of the tree. So in this tree, you can see five animals right here. So let's try to read this tree. So first of all, these two animals, leopard and lion, they come from the same ancestor, and they're actually the closest to each other in this tree, right? Because they come from right here, the same ancestor. Now, if you take this ancestor, take this non-leaf node, and if you extrapolate right here, then you can find another ancestor who actually gives birth to what is called tiger. So this means that both lion and leopard are equally distant from the tiger, and they share a common ancestor, right? And then you have the cheetah, which is even farther away. So cheetah is basically rooted with the second ancestor. So if you start from here, then you have cheetah right here, and then you go down. You have to divide even further, and then you get these two leaves, right? So cheetah actually is farther away compared to lion and leopard and the tiger, right? So basically, the leopard is the closest to the lion. It is a bit farther away from the tiger, and it is even further away from the cheetah, and it is even further away from the bear because the common ancestor for leopard and lion is this. Then you have to go another ancestor upward, then another upward, then another upward. So after going so much upward, you are going to get the bear, right? Okay. And obviously, they all started from a common ancestor. Now understand that the tree that I constructed right here, this is a hypothetical tree. That means all these non-leaf node, what I'm saying as like common ancestor, that is a hypothetical concept. Nobody actually knows exactly what that ancestor is. Okay, that is the thing. But making this kind of tree is very useful to us because then we sort of understand like how to do research on certain species and which species is similar to what. Okay. Just to clarify again, if I go to the top to bottom approach, everything started from the root. So some species was here. Then gradually it parted ways. So first of all, it became a bear. One of its like subpopulation became bears, and the other subpopulation started growing. And then that subpopulation ended right here. It again split into two. So one of the subpopulation became cheetah, which is still available. We know the cheetah are out there. And another subpopulation, it sort of went up to here, then sort of got extinct. Like it sort of took two turns. So the whole tree actually works in this way. And what like many scientists believe is that we started from this root, which is the single organism, and that organism sort of like got divided and created the current species that are out there in the world. So another thing I want to talk about is what is a species? Because I'm saying that these species are actually currently here and they are leaves of the tree. So species, basically, this is the fundamental definition. Members of the same species can successfully mate and can produce fertile offsprings, right? So, I mean, mating is not enough because lion and leopard can also sort of mate, and lion and tiger can also mate. You have obviously heard about liger, but the produced offspring is not fertile. That means the family tree stops there. Liger and liger cannot mate. That is the thing. So, two fertile offsprings can definitely produce new offsprings when they're adults, and the chain goes on and on. And so the chain has to go on. So, just being able to mate doesn't make any difference in order to be qualified as species. So, let's go next. Next, let's talk about two features, morphological and molecular. So, obviously, when you want to construct a phylogenetic tree, you need to work with some features. So one of the feature type is morphological feature, which actually deals with size, color, leg, tongue, like whatever you can see visually. That is morphological feature. But then there are molecular features. I think already understand by the name because they deal with DNA, protein, RNA, and relevant features extracted from genetic material. So currently in the modern day studies, obviously you would expect that people will be using molecular features, and that's what they do. And they construct these trees. Now, in order to construct these trees, there are various methods. Now, this lecture is about biology, so I won't I won't go into the details of these. But I want you to know about a few methods, like basically what they mean. That's what I want you to know. The first method is the statistical method, which is the most famous one, and people use it a lot. So you sort of have some model of change in features, like how the features will evolve and how they will change. Like, for example, mutation probability or morphological change probability, this kind of thing you work with here. And your goal is to maximize the likelihood of such changes when you construct the tree. This is the objective. So the advantage of this tree is it allows for confidence score. So you know how confident you are when you construct a tree, and you can incorporate complex models because you can change these probabilities as you wish or as you sort of observe, or I mean, according to the biological information. So then you have the distance based metric. So if you want to calculate the distance based tree, if you want to construct such a tree, first of all, you need to construct a matrix. So this matrix is called pairwise distance matrix. I think the name already says a lot about what this is. So if you have like three species, say cat, dog, and human, you need to have a matrix which actually shows you the distance between human to human, human to cat, human to dog, cat to cat, cat to human, cat to dog, and so on and so forth. So all possible paired distances, basically. That is a distance based, I mean, phylogenetic tree construction method. So the goal here is to construct the tree such that it best fits your distance matrix. It doesn't deviate from the matrix. Now, it is not really very recommended to use this unless you have a lot of species to deal with. Also, I mean, it cannot really deal with morphological features because if you are like dealing with size, color, etc., you cannot really get distance between those things. Like there isn't any concept of distance between those. And obviously, it's less accurate in statistical methods, but it's quite fast. Like there are methods called UPGMA, neighbor joining method. They are quite fast to work with and very simple also. And finally, we have parsimony-based method. So there is a certain assumption in this method. It seeks the tree with the fewest evolutionary changes, right? So the assumption is the entire like evolution has happened such that fewest number of evolutionary changes were made in order to survive in the environment. But again, this assumption may not be true in reality. We don't know actually. So this is something we have to be wary of. Multiple sequence alignment. Again, don't be fooled by this name. This is not a biological method. This is a computational algorithm. Computer science students sort of work with this. But you need to know about this a little bit because I want you to know about something called conserved regions and their importance in phylogenetic tree construction. That is why I'm introducing you to this. I will not talk about the algorithm, but I will just talk about what it does for you. So suppose you're working with these four species, human, sheep, rhesus, and orangutan. Obviously, each of these have its own DNA sequence. Now, what multiple sequence alignment or MSA does is it aligns all the sequences simultaneously together, right? You can see there are some like distortions. For example, G is here and all the C's are here. And then there is a change right here, there is C here, and so on and so forth, right? And there are these green regions where actually you see very less number of changes, like barely you can see any change in these big regions. So let's talk a little bit more here. So, I mean, if you want to actually understand the mutations and alterations, which you need to understand because I mean, as you saw before, for distance-based method, you need the distances. For like statistical method, you need to know what kind of change has been occurred to get the probabilities. You need to be able to like align these things together, align them together so that you can get the distance or those changes from one species to the other. But the main issue is different species can have very diverse length DNA sequences. For example, we know that humans have like around 3 billion base pair in their DNA sequence, whereas fruit fly actually has only 180 million base pairs. So you know that if you want to align a 180 million base pair and 3.2 billion base pair sequence together, it can be quite impossible sometimes. But there is a catch. There is a catch. There are some regions, such as these green regions, which remain pretty much intact across species. So there are some regions in the genome which don't really change much. I mean, even within diverse species, even within humans and fruit flies, they don't really change that much, even after all this evolution. Now, you can ask the question like why that happens. It is because the conserved regions are very crucial for maintaining different biological processes. So if you alter those, then I mean, species itself will be extinct. So you have to keep it conserved. And those conserved regions actually help to align the various length sequences or the variety of sequences together. So you know that I mean, you make sure during MSA that we align these conserved regions at least well, and the other regions should align automatically. That is basically the function of the conserved region in phylogenetics. So let's talk about evolution a little bit. So I mean, all this phylogenetics tree and everything, it is a result of evolution. So those ancestors that we are talking about, which are hypothetical, which we sort of don't know, I mean, what they are, and they are theoretical. So how did they evolve into the current species? Like, what was the process? So the first thing is we need to know is natural selection. We heard about this many times, but what exactly is it? So it is basically a process by which individuals with like good traits, better suited for their environment, survive and reproduce. Let me give a very crude example. Suppose you have a brown bear in the polar area, polar regions, right? So I mean, in the Antarctica, if you have a brown bear, it is not going to survive. Why? Because brown bear will be very easy to spot by the preys in the white ice, right? So they will never be able to catch anything. And white bears are likely to survive because they will sort of camouflage in the ice, right? So that is basically what we're talking about here. So when you have these advantageous traits in certain environments, they are sort of passed down to your children as well, and they pass them down to their children, and ultimately those fit individuals in further environment survive and the others perish. This is the basis for natural selection. So I want to give an example of bacteria. So everyone heard about antibiotic. So it's a kind of medicine that you take when you are infected by bacteria. Now, if you take too much antibiotics, you have definitely heard that you will be resistant of all the bacteria. But why is that? It is because you can see the bacteria right here. You can see two bacteria which have developed certain mutations that are actually this antibiotic resistant. So if you keep giving these antibiotics, the yellow ones will die very soon. And these few red ones which have the resistant mutation will survive, and they will keep populating. The children will again populate, and they will grow into a population of bacteria which are pretty much resistant to antibiotics. So this is a kind of natural selection. Then we have another way, which is called speciation. So speciation is the evolutionary process by which one species splits into two or more distinct species, right? So actually, like I will give an example with some bears. Let me show you this picture right here. So you know that these different bear species, they actually live in different parts of the world. For example, pandas live in like China and Asian mountain areas, where you have a lot of trees and bamboos, etc. Then we have polar bear who lives in the Arctics and the polar regions, where a lot of ice are there. Then we have the brown bear or the grizzly bears, which live in like jungles, where they need to actually hunt down prey. So based on those different kinds of regions, they have developed different kinds of characteristics. Now, all the bear basically they're supposed to come from the same ancestor, but some subpopulation of those ancestors move to different places, and after moving to different places, for whatever reason, they have to develop characteristics to survive in those environments. And through time, through ages, they became completely different species. That means if you now breed a polar bear, if you mate a polar bear with a brown bear, they may actually give birth to offspring, but that offspring or child will not be fertile. The entire chain stops there. So they are not from the same species anymore. This is called speciation. So thanks very much. This was all about evolution and phylogenetics. Feel free to like, subscribe, and donate if you want. You can buy me a coffee. The link is in the description. Thanks very much.